Just for clarification here, I was asking class what the liver enzymes are for in the Ames test, and I had totally forgotten that some chemicals or some drugs are not active or carcinogenic in their natural form, but they could become carcinogenic if they are metabolized to other chemicals. And usually the metabolism of these chemicals occur in the liver. Now we have to deal, since we deal with the damage, we also have to deal with repairing the DNA damage.
C is for clue, that's okay with me.
Now, most types of DNA damage can be fixed by the cell, and it must be fixed before DNA replication. Remember that every time we're having a permanent mutation, the permanent mutation is permanent after the cell divides, because then the side template with the mutation is going to become the parent template, is going to become the template for subsequent cell divisions. So before DNA replication, if the cell can detect the mutation, it might be able to fix it. One exception to this is in the case of transposons and also with retroviruses. Now, we cannot tell the jumping genes to jump back to the initial space before the cell divides. Right? The jumping genes jump around all the time, and then the cell is going to be able to get ready for division, and the cell cannot just stop and say, okay, all jumping genes jump back because I'm going to divide. That doesn't happen. Okay, so this is one exception to the rule. Another exception also, obviously, is the retrovirus. Once you have HIV in the cells, they're going to be there forever, as long as the cell is alive. You're not going to be able to take out retroviruses once it is integrated into the chromosome, host chromosome. Now, there are different types of damage repairs. You can have base repairs that will take care of bases that are altered. The same way you have different types of damages, you have different types of repairs to the damage. And a lot of times, one type of damage repair will act on several types of damage. So you have more or less a backup mechanism. In case one repair mechanism fails, another one will pick it up, and the cell will be able to survive. So the first one here is direct DNA repair. So this type of repair will reverse the problem at the spot, and is usually used with the thymine dimers or with the alkylated base. Now, in case of the thymine dimers, you know that thymine dimers will happen once you have UV light shining in there, and then you have the electron cloud and forms these two dimers of the T's, and now you have a problem with the DNA here. The structure is not good. The structure is just out of shape. So there is a process, there is a mechanism that is going to go there and cut only these dimers that were the problem and fix the shape of the DNA, and the DNA is repaired. So that's a form of direct repair. So those direct repairs are usually usually seen as light-dependent pathways. So what that means is that if you have, as I mentioned, abnormal covalent bonds, the protein, the repair protein will get there and will fix the problem. Now, in the bacteria, of course, we know a lot more about the bacteria system than we know about the eukaryotic system. Now, in the bacteria system, there is this enzyme called photoreactivation enzyme, or PRE, that does the same job in the bacteria as we know that is done with the eukaryotes. Well, if you look at white light, like sunlight, it has all the spectrum. Now, okay, the direct repair of the alkylated bases. If you have here, for example, a base that received a methyl group that is not supposed to be there, you have a particular enzyme that is going to go there and cut this methyl group out and restore the original base. Now look here that this direct repair of this base did not involve cutting off the base and taking out the bad base and putting in a new base with polymerase. This type of repair just happened because an enzyme went there, got the CH3 and cut it out, and now you have the base the way it's supposed to be. So that's an example of direct repair. Now you also have excision repair. Now this is not a direct repair, this is an indirect repair. That means the piece of DNA has to be substituted. Okay, just to make clear here, I don't think I was very clear here with the timing dimers. There is an enzyme here that will recognize these double bonds and undo the double bond and restore the base. Okay, I mentioned cutting out, no, that's excision repair. If you do not cut out the bases and you just restore what is in there, that is the direct repair. Okay, so we have the double bond, enzyme recognizes it, cuts out the double bonds, restore the bases, that's direct repair. Now, indirect repair, first one is excision repair. First thing that he has to do is that he has to recognize which of the templates is the problematic one. Okay, because if you have a mutation there, you know that you have one template that is not mutated and the other one that has the problem. Now, usually, when is it that DNA mutation will happen? Usually it can happen, for example, when you have replication of the DNA, and the polymerase will insert in a wrong base, and if it happens that it does not correct itself and keeps on going, then you have a problem there. Now, how is the cell going to know that that DNA is a mutated one? Okay, let's go back to replication. When the cell is replicating, you're making new DNA. How is it that the cell detects which strand is the new strand? MCM, and also methylated groups. Old DNA will have all the methyl groups that it needs. New DNA still has to get methylated. So if it has few methylated groups, it will be detected as a new DNA. So then the cell will correct a problem that is in the new DNA. We'll keep the old DNA. I think we have only about 4 slides for the repair, and then anything else that has to do with the DNA technology, with the those fancy DNA libraries and everything else that is not going to be on the test. I will not put too much on DNA repair or DNA damage. We had it last Monday, so I'm pretty sure that you guys are comfortable with it. But on the DNA repair, I send you examples of problems, right? So just get familiar with it. Also, we have seen some of these in the lab already, right? Some of the mechanisms of repair in the lab when we had the yeast lab for UV damage. So let's finish up here with the DNA repair. With the indirect repair, we have DNA mismatch repair. Now this will fix a mismatch. And now the problem is that, as I mentioned, if you have a GT mismatch over there, this is your DNA, how do you know which one is the right one? Is it the G or is it the T? Okay, now this is a problem that the cell will have to figure it out because it does not want to repair the correct base. Now here we have the common theme of this DNA damage or the DNA mutation is that once you have DNA replication, you can sometimes insert the wrong base, and then if the DNA polymerase does not recognize that to go back and redo the synthesis, does not have the backspacing activity, right? 5' to 3' exonuclease activity, or 3' to 5' exonuclease activity. So if it does not do that, then the mutation is inserted, and now the cell has a problem. So for this indirect mismatch repair, what is it going to do? First, it's going to see that one of these strands, usually the one that is just made, the newly synthesized strand, is the one that has the problem. So here, the indirect repair is very biased. It doesn't even ask the strands which one is it. It'll just assume that the new one has the problem. It's pretty much the same type of dynamics when you go and you work in a new office, and then something happens and everybody blames the new guy. It comes all the way down, all the way from the DNA, the type of mentality. Okay? So the new guy has the problem. So the other hint here is that how do you recognize the new guy? Because they both look the same here. There are two strands of DNA. Which one is the new guy? Well, the new guy is going to know less than the old guy. Is going to have less stuff in that office. Is going to be less methylated. Is going to have less modifications. Is not going to be attached to those. Which one of those? Disney World type of park? Not MGM, something else close to it. MCM proteins. I meant a Disney park. You have to know that Disney parks. Universal is universal code. No. No. When you recognize new strands and old strands, when the cell is replicating, it has to know which one is the new, which one is the old. It does not want to replicate the strand twice. And it has to recognize in order it has to make some repairs on the strands. So the one that did not get methylation, look here, in this case is the red one down here. The DNA, the repair system here will recognize that as the problematic one, the new guy in the office. So it's going to break it in pieces and reconstitute it. Now, this is also important to know here. The time that this DNA is not methylated is a very short time, because right after replication, you know that the methylases are going to go there and start making this strand just as the parent, just like the parent strand. Yes.
Which one was MCM?
Which one do you think? No, the new guy is the one without the resources. So the old guys are the one with all the stuff in there. They have the methyl groups. Okay, so here, old guy has all the stuff, the new guy is lacking. So they recognize missing methylation here. So they say it's unmethylated for a short period, and it can also be called as hemimethylated. And then, of course, if you wait too long to repair this problem in the DNA, then it will not be able to repair anymore, because then all the DNA will get methylated, and then it's not going to know which one is which. Okay?